Hello everyone. Today, we're diving into a fascinating topic called the importance of primary structure and how it determines higher order structures. By the end of this lesson, you should be able to explain what primary structure means, describe how it influences protein folding into higher order structures, and understand the serious consequences when the structure is altered. To kick things off, let's do a quick activity entitled Send Help. I'm handing out a strip of paper to each pair. On it, you'll find some jumbled terms. Your task in your pairs is to number these terms from 1 to 4, arranging them in the correct order to show how the primary structure of a protein is formed. You have exactly 30 seconds to complete this. Go. Time's up. Alright, who would like to volunteer to share their sequence with the class? Excellent. The correct sequence is amino acid, peptide bond, amino acid sequence, and primary structure. Now pause and think about that for a moment. The perfect sequence is crucial. So what happens if that sequence, the precise order, becomes distorted or changed? Keep that thought in mind as we move forward. To begin our main explanation, I want to hear your thoughts. What do you think the primary structure of a protein means? Great thoughts, you're on the right track. The primary structure is indeed the specific sequence of amino acids in a polypeptide chain. Think of it like a unique string of beads, where each bead is an amino acid, and these amino acids are precisely linked together by peptide bonds during protein biosynthesis. The chain has two ends, an amino terminus and a carboxyl terminus, and it uses 20 different amino acids in various combinations to form a unique sequence. But what happens if just one amino acid in the sequence is changed? That very question is the core of our discussion today, leading us to our main topic. Now, let's dive into how this primary structure influences everything else. Can you recall what fundamentally holds proteins together in their basic chain form? That's correct. Peptide bonds link the amino acids in the primary structure, but beyond that, proteins start to fold in more complex shapes. The secondary structures are composed of recurring folding patterns of the polypeptide backbone itself. This polypeptide backbone is directly influenced by how the primary structure determines where and how these specific patterns form within the polypeptide chain. Now, do you recall what type of bonding is primarily present in secondary structures that helps create these patterns? You've got it. It's hydrogen bonds. As you can see here, the two most common secondary structures are the alpha helices, which are like a coiled spring, and beta sheets, which resemble a pleated folding paper. Now, as you watch, observe how these basic patterns emerge. Next, we move on to an even more complex level of organization, the tertiary structure. Tertiary structures are the intricate three-dimensional foldings of a single polypeptide chain. This is where the protein truly gains its unique overall shape. As you're watching, try to recall the different types of bondings or interactions that help stabilize a tertiary structure. What are some of those forces? And finally, we have the quaternary structure. This is an arrangement of multiple polypeptide chains, what we call subunits, coming together. The specific arrangement of these subunits is critical for their overall activity and function. Those interactions you just mentioned for tertiary structures are exactly what we call intramolecular interactions, and they make these complex structures stable. We have hydrogen bonds, hydrophobic interactions where nonpolar parts hide away from water, ionic bonds between charged groups, and strong disulfide bonds that act like molecular staples, all playing a role. Now let's tie it all back to the primary structure. The primary structure dictates the exact sequence and positioning of all these diverse R groups. Remember, Those are the unique side chains of each amino acid. These specific arrangements of R groups determine how the protein folds at every single level, from the local secondary structures right up to its complex tertiary and even quaternary shape. To summarize all of this, think about how an origami is folded. You start with a flat piece of paper. That's our primary structure, the linear sequence of amino acids. The way that you make the first folds, our secondary structures, then directly influences how you can make the next, larger folds, our tertiary structure. And if you have multiple folded pieces that fit together, that's our quaternary structure. Every fold depends on the previous one, and it all starts with the initial, flat paper. Just like proteins, their higher order structures are subsequently determined by that initial amino acid sequence. Alright, let's move on to the real-world impact of all of this. We often hear that form follows function, but how does this principle specifically apply to proteins, the molecular workhorses of our cells? For a protein to perform its biological job, it must first achieve a precise, unique, three-dimensional shape or conformation. This native shape is absolutely essential, truly analogous to a key perfectly fitting to a lock. If the shape isn't right, then the function can't happen. Within the cell's incredibly crowded environment, there are chaperone systems that act as molecular guides, helping proteins achieve their proper structure. So, why do you think that these helpers are so vital for our cells to function properly? This proper folding process is fundamental for maintaining cellular homeostasis, the stable internal balance vital for life, and for ensuring our cells, and ultimately our bodies, function optimally. So, if a shape is everything for function, what ultimately determines its perfect shape? It all starts with the protein's primary structure, its unique linear sequence of amino acids, which serve as the ultimate blueprint encoded by its gene. Can you recall what the primary structure of a protein refers to? Exactly. Now, let's shift to the consequences. What happens when that critical blueprint, the primary structure, gets altered? It's explained that mutations in a protein's gene, perhaps from environmental factors like UV radiation, or errors in a DNA replication, can change just a single amino acid. Can you recall how the primary structure of a protein is initially formed? Right. The alteration in a primary structure is like having a defective instructional manual. This directly interferes with the intricate protein folding process, leading to a misfolded protein. This misfolded protein cannot achieve its correct native conformation, resulting in a loss of function. Though our cells do have degradation pathways for quality control, attempting to identify and eliminate these improperly folded proteins. However, the complexity of protein folding means errors can occur, and sometimes misfolded proteins evade degradation and accumulate. So, if a misfolded protein isn't removed by the cell's cleanup crew, what kind of problems might it cause in the cell or even on the whole body? The accumulation of misfolded proteins has profound clinical significance, underlying many pathological conditions from neurodegeneration to even certain types of cancer. Before we delve into specific examples, I want to hear from you. Can you recall why proteins are broadly considered essential for life? Precisely, they do virtually everything. A powerful illustration of altered primary structure's impact is sickle cell anemia. Here, a single amino acid substitution from glutamic acid, which is changed to valine, in the hemoglobin beta chain due to a single base chain in the DNA causes hemoglobin to form rigid fibers. These rigid fibers distort the red blood cells into a sickle shape, explaining how these rigid cells clog blood vessels, leading to chronic pain, organ damage, and reduced life expectancy. This really shows how one tiny change in the blueprint can have devastating effects. Now, for a fun way to review and summarize our entire discussion, we're going to play the protein chain reaction. Here's how it works. I'll hand an object to one of you. When it's your turn, you quickly add one word or a short phrase that summarizes something important we've discussed today about protein structure, folding, or its consequences. Then you quickly pass the object to the next person. We'll keep going, building a continuous sentence or an idea until the object goes through a few rounds. The goal is to see how much we can collectively summarize in a quick, fun chain. Alright class, as we wrap up, let's revisit the core idea that was woven through our entire lesson today. We've talked about how incredibly important the structure of a protein is for its function, and how complex that folding process is. So, to summarize the absolute key takeaway, what specific part of the protein ultimately determines how it folds to all those intricate higher order structures? Secondary, tertiary, and even quaternary. That's right. The primary structure is truly the master blueprint. This single linear sequence dictates every twist, turn, and interaction that leads to a functional protein. And a fault in this blueprint is what can lead to devastating consequences for our cells and for our health. Understanding this fundamental concept is crucial to grasping how life works at a molecular level. To make sure we've got a strong grasp on these fundamental ideas, let's do a very quick summative quiz. Just three multiple choice questions to test your understanding from today's lesson. No need to write this down, just think of your answer. Thank you for all your excellent participation and engagement today. You did a fantastic job. Goodbye class and see you around.